All right, it's my pleasure to introduce our first speaker of today. Ethan Woodyard is a PhD student at Mississippi State University's College of Veterinary Medicine and advised by Dr. Matt Griffin. His dissertation work focuses on mixozoans of importance to catfish aquaculture, in particular Hanagaya telluri. However, today he's going to present on a project that he started as an undergrad at Mississippi State's College of Forest Resources. Looking forward to hearing about the alligators.
All right, thanks, Dr. Phelps, for introducing me. So I've got this title with a bunch of buzzwords. My hope is that maybe YouTube will make this the thumbnail for the video. Maybe we can drive some more clicks. But anyway, here's what I'm actually doing. Is just we're looking at some parasites in alligators and how the lesions associated with them might be creating portals of entry for secondary infections with bacteria. Full disclosure here, I'm running on about 2 hours of sleep, so if I drop dead, please redirect your inquiries or comments to my co-authors who are listed here. I want to thank Lori Ford and Divya Rose for handling a lot of the bacteriology stuff, because that's not really my wheelhouse, but that was essential to this project. Dr. Baumgartner for handling the histo, Dr. Rivers for being a wizard on the SEM stuff that you'll see later. I can struggle with something and he comes in 4 seconds. It's the best images I've ever seen. Scott Rush for helping us get some of the alligators, Celine Slivka for helping us with a lot of the alligator necropsies, Jonah Wynn for doing some of the sequencing PCR work, Matt Griffin for being my advisor and just kind of supporting me all around and giving me the freedom to explore things that are kind of outside of my dissertation work to get kind of a broader perspective on things. Our librarian, Derek Marshall, for getting us some of the references that are used in here. Some of these Chinese papers and stuff are pretty hard to get a hold of. He really came through on that. And then Graham Rossa for being around for pretty much all of this project and helping with pretty much all of it. So I also want to thank Anna Phillips at the Smithsonian for sending us a lot of voucher specimens of these trematodes to examine, and the alligator processors, Woody and VC Kane, for helping us get most of these alligators. This project would just not have been possible without them. And then I got a couple funding sources listed here. Again, this work would just not be possible without these people. So before I get into all of that, kind of why we care about alligators, of course, there's conservation reasons, but there's also a lot of aquaculture of alligators. You can see all these countries that export alligator products. Kind of a surprisingly widespread industry, at least surprising to me. Unfortunately, it's kind of hard to put a dollar value on it because while these countries report, number of skins exported, they're not necessarily obligated to report a dollar value for those. And as y'all know, with an aquaculture product, the prices can fluctuate all the time. So it's hard to really infer a dollar amount from just that information. But you can see several $1 million industry here. And I'm going to be talking about trematodes. So I think it'd probably be a good idea to just do a quick review of trematode life cycles. In our case, you've got this alligator definitive host, and this has the adult trematodes in this host. The trematodes release their eggs. Those are released into the water. Those hatch into Myricidia, which are infectious to a snail, first intermediate host. And that snail is going to release these Cercaria into the water, which are infected to a fish, second intermediate host. And they're going to develop in that fish into this metacercaria stage. For this intermediate host, it's in the gonads. And then that fish is going to be ingested by the definitive host. The metacercaria is going to mature into an adult, and the life cycle continues on like that. So this project kind of got started, like Dr. Phillips said, I see it in my work in wildlife and fisheries. In 2016, we started doing a survey of alligators in Mississippi to look at their internal parasites. And where we got most of these is from the canes that you see up here on the upper left. Those are the alligator producers I mentioned earlier. So we go down there, and this is what the end product of alligator processing is. Now, the hunters that hunt alligators in Mississippi bring their catch to the processors. The processor removes the skin, this meat off the tail and the head. That's really what people want. from their alligator hunting, besides just the experience of doing it. But they leave behind all this, which is this parasitology gold mine year. So we bring those back to Mississippi State. We separate out the organs, and just to kind of see down here, this is that skin I was talking about. That's kind of what the hunters want to take home with them. And we recover parasites from the GI, but we also on the tongue recovered these trematodes. You can kind of see circled here. Pulled those off, put them in saline. They're still alive when we get them. So we heat relax them, what we call it, in saline, and that just kind of, you can see they flatten out, and we do that so that we can mount them on slides to do morphological characterization of these trematodes. So we did that a few years back and did the first molecular characterization of this trematode, Odneotrema incomodum. And we know it's that because we compared to all this morphological data from previous accounts. And we just kind of plug that into some phylogenetic analyses. And what's kind of cool about this is you see all these genera, these infect fish-eating birds, and then there's this alligator infecting one that seems to be like basal to all of these. So we're not really sure what that means for the evolution of these parasites, but maybe they were in some kind of shared ancestor and then diverged off, and the parasites diverged as well. I do kind of want to point out the original description from way back when, not a lot of data there, and we'll kind of get into some of that a little bit later. But that data, that sequencing data we deposited, we kind of kept using that for subsequent papers to characterize some other trematodes and just kind of add some more to what we know about the evolutionary relationship between these parasites. And we are comparing that molecular data to systematics that's kind of based on what we know about definitive hosts and to kind of the morphology, what we know about life cycles in general. And for this group of trematodes, we've kind of got most of the genera represented. You can see there's still some missing. So over the course of these studies, we've kind of accumulated a lot of these specimens. You can see like in this green, that's our specimens from the studies so far and then the present work. And then down here on the right in orange, you can see this is a couple of specimens I got from a museum. They're not from a published study, which is a shame because they look much better than mine. But anyway, I also got these metacercaria that are reported as either Odneotrema incomodum, or they're reported from hosts that Odneotrema might use as a fish intermediate host. Incidentally, none of them are even close. And then I want to really brag about this one. This is one of Lady's type specimens from 1856 that was sent to me from the Smithsonian. But before I do that, I just want to point out, this is all the known slides of this. Before we started doing this work, there were actually more papers on Odneotrema or addressing it than there were specimens. But anyway, the specimen of Lady's, there was a slide, but it was lost in about 70 years ago. So all that's left is these worms in vials. Surprisingly, the Smithsonian was willing to send me one of these to make my own slide of, and that's kind of what this guy is down here. But anyway, we did a morphological and molecular characterization of all these things over the last few years. And the way we've been doing that is with what's called hologenophores. And what that is a worm that we take all these morphological data from, but we also cut a little piece from the same worm that we get the morphological data from. And we extract DNA from that and do sequencing. And we did all these regions from 13 of these worms. And we put all that data into phylogenetic signal assessment. And this is a lot. I really should probably like pare this figure down. But it's just, you're looking at a phylogenetic analysis of all of these trematodes in this this Clinostomidae. And what we're trying to look at is there's all these different regions that people sequence. It's kind of inconsistent between studies, and it's not really clear or quantified what regions are better for resolving which sorts of taxonomic relationships. So what we were trying to do here is see if you were trying to work out a particular evolutionary relationship or just identify these things, what region should you use? And without like trying to interpret all of this, the real takeaway here is that the cytochrome oxidase barcoding region is doing a great job for separating things at the species level. But these ribosomal regions that are not quite used as commonly you can see that signal is much higher for these earlier relationships. So if you're trying to work that out, it might be better to also include these regions that are not used quite as commonly. But kind of getting back into some actual aquatic animal health stuff, I think y'all are probably kind of interested in that more than trematode systematics. We did this work kind of recently where we were looking at some actual histopathology on alligators infected with this trematode. And you can see there's some pretty gnarly lesions here. There's like one of the trematodes still attached. You can see these fibrotic lesions associated with the trematode. And there's some like necrotic mucosa happening here. And I just want to point out, this is what the pathologist is telling me. So I trust his work, but no hard questions about that, please. So you can see there's inflammatory foci with the bacteria, and that's just associated with these lesions. You can also see an ulcer with these same bacteria, and then just a site of trematode attachment. So I had this great idea that there would be, we would look at these bacteria associated with trematodes, and just these bacteria in alligators generally. Turns out, like a lot of great ideas, it's been had and done a lot before, a lot. So what can we actually add to this to kind of save this project? What hasn't really been looked at is how the bacteria are getting into the alligators. It's kind of speculated that, for example, like when they're in raceways, maybe they're getting their skin abraded on the concrete, and maybe the bacteria get in that way. But it's kind of all speculative. And what's also missing from a lot of this work is a molecular identification of the bacteria, and the diversity of those is just, y'all are aware, is just getting more and more complicated. It's apparent that molecular data is just a requirement at this point. So that's what we hope to add. But to do that, we need to collect some more alligators, because before we just had histo, we didn't do any culturing of the bacteria. So we can see that there's something that looks like bacteria there. We can't really ID them. So here's us collecting more alligators. It's me, Celine, and Divya being grad students. There's one of the alligators being dead and being an alligator. And we also collected some of that tissue for SEM, and you can see there's one of the trematodes attached here, and there's one of the lesions associated with them. And if we kind of zoom in on that, you can actually see the lesion looks just like the oral color of the trematode. It's pretty cool to me. And if we zoom in even more on that, we can see these look like bacteria. They could be Aeromonas, but it's not really a definitive ID from SEM, but it does look pretty cool. And it's just another line of evidence that maybe when we look at these lesions, there seems to be bacteria associated with it. But we also did some bacterial cultures. There's Divya here culturing some of these bacteria on plates. We took swabs from the tongue, some from the lungs, and also some from the cloaca, and cultured several Aeromonas species, mostly Veronii, but then from the cloaca, we got some Veronii and some Ducensis. But we put the Veronii that we have so far into a gyrase B phylogenetic tree with Mr. Bayes. And you can see the tree's doing a pretty good job of separating things out by species. I mean, there's some weird polyphyletic things happening down here, but for the most part, it's doing a pretty good job with decent support values. And it's putting all of our Veronii in a clade with pretty good support. If we zoom in on that, I was really hoping there would be like, this is the alligator isolate of Veronii, but that's really not what's happening. It's just kind of grouping with everything else. So this work has been done. There's been some sequencing. If you look back at this, there's all these alligator Mississippiensis of ours, but then there's this alligator sinensis. What's that about? Well, this work has actually been done a lot with Chinese alligators, but the work was done in Chinese, so I didn't know about it. But if you look at these, the titles, you can see that's a lot of what they're doing. Unfortunately, there's not a lot of sequence data available for those for me to compare with ours, but I am in contact with some of those authors, and I am working on getting a hold of their sequencing data. So that's kind of ongoing. I might want to do some additional sequencing on the Aeromonas species from this paper, maybe Illumino, or maybe just throw in a couple more regions to do a proper MLST. We've got histology on some more recently collected alligators that I want to incorporate into this, just to show again the histology associated with SEM and with the sequencing data to match up with what we did before. And I also, there's parasites in the lungs, these pentastomids. I saved some lungs for SEM, but I didn't do it in the best way. I just kind of cut out these sections, but what I really should have done is filled the lungs up with formalin to let them fix, and then opened them and cut out just to retain some of the like finer structures that you don't really see as well as you could in the SEM that we do have. So that's kind of future directions for this stuff. And with that, I'm glad to take any questions.
All right, thank you very much, Ethan. Interesting talk. Folks, feel free to unmute yourself or drop question into the chat box. We've got time for a few. Oh, Ethan, you got a question here in the
Oh,
chat.
yeah. I see one in the
For
chat
clarity
here.
on where the specimens came from.
So these alligators are all wild caught. We have a hunting season now in Mississippi, and they get permits to go out. Mostly this is in the southwest region of the state, but these are all wild caught. I would really like to get a hold of some farmed alligators, but that's not happening so much in Mississippi. So we've got to deal with getting permits to export samples from farmed alligators from like Louisiana or Florida, where that's really happening. Hopefully that answers your question.
Yeah, go ahead, Rod.
Great talk, Ethan. I might have missed it, but are the Aramonas, are these actually causing mortalities in the farms? Do you know?
I don't, yeah, there are some associated with mortalities in farms. I don't know off the top of my head how widespread that is. It's not really something that people report prevalence on, as far as I know. It's usually just like, hey, look at this weird thing that happened. It's kind of a one-off case report type thing.
Thanks.
And I see I've got another one in the chat. Are the parasites carriers of the bacteria? We did swab some of the trematodes themselves to see if they had the bacteria on the surface of them, and we got the same thing back. I did actually try on whole worms that I had rinsed and extracted DNA from to see if I could, I screened those with the bacteria primers, and I was not able to amplify anything from those. So I don't think that the bacteria are in the trematodes, but I think what's happening is these Aromonas species are just present in the environment, just ubiquitously, and the trematode is creating a lesion, and then the bacteria are just kind of opportunistically getting in that lesion. Let's see, I'll get another one. Were they fresh or were they preserved before they came to you? Wondering if it matters how long the animal is deceased compared to how well you see your parasites and bacteria. So they were all collected fresh within about 24 hours of being killed. They're put in a cooler. That could be an issue if the bacteria are like growing more on the dead alligators. It's kind of something I'd like to address in the future. Just like logistically, a few alligators come in at a time, and we get a call from the producers, and we've just got to drive over there and pick up as many as we can. But it's kind of unpredictable when they're going to have them come in. I'd really love to go kind of stay out there where they process them and collect these samples as they come in to get them as fresh as possible to kind of address that. But yeah, that is definitely a limitation of what we're doing here.
Great. Any other questions out there?
Do you see seasonality with presence of the trematodes, warm or cold weather? And do we see trematodes in most samples? The trematode prevalence is about 60%, but as far as seasonality, that is definitely something I would like to look at. But because we're relying on the alligator hunting season, that's only a 10-day season in Mississippi, and it's in late August, early September. So we're really restricted to that time point of when we can look at these. There are some opportunistic collections when nuisance alligators are dispatched outside of those times, but it's kind of hard to get a hold of somebody in time to go get that one alligator, depending on where it is in the state. So it's something I would like to look at, but it's just like with our opportunistic kind of collection we're doing through processors, it's not really feasible for us.
Great. Well, thank you very much for your presentation, Ethan.
Well, thank you.
A virtual round of applause for you.